So hi everyone, my name is Cindy and I am a second year master's student here at York University in the Rehan Lab. And my research looks at wild bee microbiomes in an urban context, particularly in the city of Toronto. So microbes, including bacteria, fungi, protista, viruses, all these tiny little organisms are often found within or on hosts such as bees. And so looking at the microbiome, we examine all the microbes on a host, and in examining the complex and dynamic relationships that a microbe has with a host, we are examining the holobiont. A lot of these studies characterize something called the core microbiome, which consists of all the microbes that are commonly found in a species. So for example, we know that lactobacillus is commonly found in the human gut, and the same thing is actually true for bees as well. The role of the microbiome in bee health ranges because symbioses can range from being negative to neutral to beneficial. However, oftentimes microbes are actually beneficial to bee hosts, improving bee immunity, helping against pathogen defense, aiding in nutrient utilization, aiding in metabolism, reducing metabolic toxicity. The list goes on and on. And so we know that altering these microbiomes actually results in decreased fitness. Oftentimes, if you sterilize the microbiome, you end up with negatively affected growth rates, biomass, and survivorship. So microbiome research in characterizing the microbiome often looks at two things. Firstly, composition and diversity. Composition characterizes the types of microbes present and their taxa, saying what kinds of bacteria, what kinds of fungi are present, whereas diversity is a metric describing the number of different microbes that are present. Another way to look at the microbiome is to examine correlations. So seeing if the presence of one microbe predicts the presence of another, or if the absence of one microbe has a negative effect on host health or with the other microbes in the microbiome. A lot of research questions for the microbiome are related to how it is acquired and established and maintained in the host. Oftentimes, social bees are able to maintain a core microbiome that is inherited through vertical transmission, so going from mother to offspring, or through social interactions. Solitary bees don't have these social interactions, and so they rely more on the environment to acquire and obtain their microbiome. Things like foraging or from their nest environment, it's less about social interactions and more about what they are exposed to. And so this means that changing environments are particularly important in solitary bees in affecting the microbes and how that dynamic relationship with their host is. In growing cities that are adding impervious surface, land use development, and affecting green space availability, also with climate change occurring because of urbanization, there are many different ways in which the environment is changing for our bees. And with urbanization being an increasingly worsening issue, such as in the city of Toronto, where it's always expanding, growing, and developing, we know that different ways in which urbanization affects the land, such as through natural habitat, floral resources, and bee species richness, have been shown to affect the bacterial composition in Osmia lignaria and in bees in general. So with wild bees being exposed to varying environments and pesticides in both agricultural and urban contexts, my research looks to see how those environmental factors are affecting the microbiome. Our study species is Ceratina calcarata. It is one of the small carpenter bees and commonly found in North America and particularly in this region of Ontario. It is a stem nesting bee and subsocial bee, which means that it exhibits maternal care of offspring. So each mother provides a pollen provision in each brood cell, laying an egg, and so that pollen ball will sustain that larva all the way through its development. The aims of my research project are threefold. The first is to characterize the microbiome across developmental stages, going from larva to pupae to adult. Then to characterize bees across Toronto, across an urbanization gradient, examining variables including land use development, green space availability, annual temperature, and annual precipitation. And thirdly, to examine how urban and rural bees compare. We currently have sites set up at farms about an hour away from Toronto, and so we're comparing urban and rural bees. Our methods are similar across all three aims, but our first step was always to bring back Ceratina calcarata. We have collected them in the wild, just in parks and in our areas downtown in Toronto, as well as, again, setting up sites on the farm so that wild bees can create nests. And so once we bring back the nests to the lab, we are then able to separate pollen provisions, brood, and adults, freeze them, perform DNA extractions on them, and then send off that DNA for sequencing. We perform amplicon sequencing so that we are doing metabarcoding such that we target specific regions on the genome that are then we can relate those reads back to different taxonomic classifiers so that we are able to say what kinds of bacteria, fungi, and plants are present in our samples. And then across the 3 different aims, we were then able to characterize the microbiome across the different developmental stages, across different urban landscapes, again, looking at the variables of land use development, green space availability, temperature, and precipitation. And then that rural and urban comparison. We're also currently doing work on pesticides to see if there are pesticides in the bees in the farms as well as in downtown Toronto, and then characterizing the microbiome based on that. So I'm going to begin with some of our results from our developmental microbiome study. And what we found was that microbial diversity, both alpha and beta, differs between particularly adult and juvenile stages. So here I have a figure with the Shannon index, and a greater value for a Shannon index indicates increased diversity. So with a lower bacterial Shannon index found in adults, we believe that Ceratina likely benefit from having a more diverse microbiome in earlier stages of development. However, overall, there is still a concerning lack of diversity where sometimes samples would only have 1 to 2 bacterial or fungal genera present. And then this causes low abundances of certain important genera, such as Apolactobacillus, formerly Lactobacillus, which we know is a common symbiont in wild bees, and particularly in Ceratina calcarata, having seen that in previous studies and in different locations as well. Through the study, we also found that there are both positive and negative correlations between bacterial and fungal genera, meaning that the presence of one could predict the other. But also interestingly, many of these fungi are thought to be plant pathogens, which kind of explains how they are sort of acquired from the environment. As well, some of these bacterial symbionts are able to inhibit fungal pathogens, which suggests that there is some beneficial relationship that these fungi are having for our bees. Moving on to our study of the microbiome across an urbanization gradient in Toronto. This is the map of our collection sites from Toronto, with each circle representing a collection site. A lighter color such as white indicates a lower level of land use development, which we call Dev 1, ranging to Dev 5, which indicates an increased amount of impervious surface or land use development. And in this study, we found that the relative abundances of taxa differ across different levels of impervious surface. So for example, in orange and in gray, we have Acinetobacter and Sphingomonas, which are much more relatively abundant in areas with lower land use development and much less abundant with increased amounts of impervious surface. Again, you can see that there is an overall lack of diversity as many individuals or individual columns show only 1 to 2 different colors, which indicate only 1 to 2 different bacterial genera present. We were also able to perform RDA analyses, which are similar to APCA, but also include the different environmental factors. And what we found was that land use development and green space are associated with bacterial taxa and those relative abundances across individuals, and that green space and precipitation were associated with fungal taxa. So in this way, we see that the different environmental factors are pushing and pulling and affecting the microbiome in different ways, such that there are many different factors that we have to consider in how that affects the composition of both bacteria and fungi. Again, we were able to perform correlations analyses to see if there were relationships between bacteria and fungi such that one could predict the presence of another, and breaking it down into individuals of different impervious, from the sites of different impervious surface, we found that some co-occurrences are more common in areas of specific sites. So for example, the relationship between Acinetobacter and Sphingomonas is found in areas with moderate to high land use development and not so much in areas with low land use development. So what this means for the bees is that the environment might be playing a key factor in shaping the bee microbiome. Adult bees tend to differ from larval and pupal bees, which could be a factor such that they are able to interact with the environment and that they're exposed to such. But also our most significant environmental factor was land use development, although green space, precipitation, and temperature all played a role as well. Diversity seems to be relatively low in urban bees. Again, overall, you can see that the composition in each individual is sometimes limited to 1 to 2 genera. And so that is sort of concerning because we know that there are oftentimes benefits to having a diverse microbiome. Missing key taxa, such as Apelactobacillus, raises the concern for bee health, as the underrepresentation of beneficial taxa is concerning. However, we do need to do more functional work to sort of be able to understand all the microbes' role, because it's not just Apelactobacillus, there's a whole range as well. And so a lot of functional work needs to still be done. And that sort of leads me off into future directions. So again, we are currently studying those bees from agricultural sites, so those rural bees from the farms, and we are going to be characterizing those against the same environmental factors: impervious surface, green space, precipitation, and temperature. We have also sent those off for pesticide analyses. We obviously know that pesticides have a negative impact both on bee health, but also that it affects the microbial composition and diversity as well. So as we learned from this morning, with changing pesticide regulations in both agriculture and in urban settings, we have started to characterize that to see how that is affecting the microbiome. Again, functional work needs to be done to examine how microbes are affecting host health, with experimentation specifically manipulating the presence or absence of and comparing the abundances of certain taxa being interesting, so that we know what it is that these microbes are actually doing to bees. And a lot of work in our lab has been done to characterize wild bee microbiomes in general, as wild bee microbiomes have been shown to differ from honeybees. So currently in our lab, we're working across many different species and across many different landscapes all over Canada, including species such as Agapostemon and Megachile, Anthidium, Hylaeus, and across the different provinces such as Ontario, Quebec, and Nova Scotia. So a lot of work still needs to be done on characterizing bee microbiomes. But for today, I'd like to thank you all for listening, and thank you to everyone in the Rehan Lab for all of their help and guidance, and thank you to all our funders. And I believe I have time for any questions if anyone has those. So the question was, if I had to speculate, what would be the, sorry, what was that? So the functional relationship between diversity and land use change. I think with different land use change, with urban development, we have lost a lot of bacterial diversity, I believe. And so that is just impacting all of the beneficial. So we're lacking all of the beneficial microbes such as Apelactobacillus in urban centers. And so that's what I think is sort of concerning, that we're losing a lot of bacterial diversity. Clement? Yes, particularly in honeybees, we know that for sure, but it has been characterized in, I believe, in osmia, in nomia, and in bumblebees as well. So you mentioned that maybe some soldier bees get mainly microbiome from the environment, but given that they're still being provided pollen, like long and pollen from a high microbiome diversity, kind of that indicates that they do get something from kind of a vertical, kind of semi-vertical condition. Yeah, but it's also the question was if it is if brood are having access to pollen, which is inherently part of the environment, but also from the mother, is it a form of like vertical transmission? So because the pollen itself, the microbiome is differing between environments, so between mothers it differs, depending on where or what flower that mother foraged from that day. We did find that the microbiome changes across the pollen provisions itself. So it is a form of vertical transmission, but it's dependent on the environment. Oh, yes. I don't have the sequencing data yet, but we do have some pesticide results back, and we did find some neonics at some of the farms, and so different pesticides, yeah.